So I'm going to continue the story that Annie started and really focus on from the lens of the microbial community. So I'm hoping to convince you that although microbes may be tiny, they play a really large role in the puzzle that we're looking at here. And I also want you to keep in mind that although I'm talking about nitrogen, think about this at the broader scale, how microbes may respond to other coastal stressors as well. And so as Annie covered, we know that nitrate can play two different roles, either as a nutrient in primary production, which can help organic carbon storage, but it also can serve as an electron acceptor, fueling heterotrophic microbial respiration, such as denitrification, in which case this may result in a CO2 release. And so to try and explicitly test that, we ran a very controlled experiment in the lab where we collected sediment from tall Spartina alterniflora up in Plum Island, sectioned the sediment at different depths, ranging to about 100 years worth of sediment accumulation, and separated these sediments into two treatments, a nitrate treatment and an unamended kind of control, seawater control, so conditions that we would normally see in a salt marsh. And these little reactors are useful because you can very carefully manipulate what's going into them and coming out. So we can specifically look at the response of the microbial community here. And so the first thing I'm going to show you is dissolved inorganic carbon production, so what's going into and coming out of these little reactors. And so DIC is a good way of looking at total microbial respiration of the system. Yellow is the nitrate treatment, and blue is the control. And so all I want you to see here is that over time on the x-axis, yellow, so nitrate is stimulating respiration at all these depths, even the organic matter that's much older, okay? So again, because nitrate is a favorable, energy-rich electron acceptor, it's stimulating respiration of the microbial community. We also know that nitrate, again, can be used in different ways. And so what I'm showing you here is similar to the DIC graph, but instead nitrate consumption in each of these depths, just for the nitrate treatment. And so we can see that these curves very closely match that of DIC production across all the depths, but from this we can't really tell how that nitrate is being used. So what we did is we added nitrate in the form of N15 so that we could track the fate of that nitrate into different things such as denitrification, which ends in N2 gas, and deNRA, which ends as ammonium. And so here I'm showing you overlaid on those nitrate consumption plots, denitrification in the blue bars and deNRA in the green bars. And from this, the fact that they kind of very tightly overlay, it suggests to us that a lot of this nitrate is indeed being used as an electron acceptor and is changing the respiration of the microbial community. And so naturally, the next step is like, okay, fine, what's happening to the microbial community? Are we changing the microbes entirely? Are we just changing what the microbes are doing? So we can answer that in several different ways. So the first thing I'm showing you is sequencing DNA of that sediment, okay? So this is just sequencing one gene, the 16SR RNA gene that all bacteria have, and this gives us the taxonomy of the system, okay? And so in this PCOA, essentially every point represents a sample community, a sample microbial community. And so all I want you to see is we have the unamended control treatment here, but when we add nitrate, we're fundamentally altering the microbial community composition. Okay, so now you're like, okay, Ashley, fine, you're changing the microbial community, but what are they doing? The next thing we can look at is using what's called shotgun metagenomics, which looks at all the genes in the system. So thousands and thousands and thousands of genes. All I'm showing you here are along the y-axis are just very broad categories within which these genes are categorized. In this case, I switched colors on you, but the green is the nitrate treatment. And the big thing here is that when we add nitrate, we are changing the genes involved in nitrogen metabolism. We're drastically increasing the abundance of those genes. Okay, so we're changing the microbial community, and we're changing it by increasing the abundance of nitrogen-related genes. So circling back to how this relates to the processes that we measured that I talked about earlier, we did a correlation between those processes as well as various genes related to dissimilatory nitrogen cycling, so stuff like denitrification and deNRA. I removed the names of those genes to spare you, but trust me, they're there. And so basically, everything that's in red suggests a positive correlation. Anything in blue is negative. Anything that's bolded is significant. So the big takeaway here is that as we're seeing an increase in that DIC production, denitrification, deNRA, we're also seeing an increase in those specific nitrogen-related genes involved in nitrogen cycling. Okay, so I know I went through that quickly, but really the main thing I want to show you here is that we shifted the microbial community by changing the genetic capacity of those microbes, okay? This resulted in stimulating denitrification in DNRA, accessing more complex organic matter, and then overall degrading more organic matter. And so this obviously plays a piece in the puzzle of this blue carbon that we're looking at. And so we really need to consider the role of the microbes when thinking about these things, because as fast as plants respond, microbes respond even quicker than that. And I will argue that, you know, microbes they're hard to study, but sequencing is becoming cheaper and cheaper and quicker. And so maybe we need to think more about them. I also want to put in a quick plug that I'll be starting a project with collaborators in early 2020, looking at specifically the microbes that are responsible for degrading carbon in salt marsh sediments. So if you have any salt marshes in New England that you think might be a good location for me to include in the study, I would love to talk to you, because I'm pretty much starting from, you know, from ground zero in a way. Not really. You guys are helping me out. But I won't be able to stick around in the afternoon, so if you can come find me at lunch, that would be great. Thank you.
Thank you.